Humans have been changing farm animals for thousands of years. We've changed their colour, size, and a few other things. This is so that they produce more meat or milk, or are better able to thrive in particular environments. All this has been done through selective breeding. By selecting and breeding individuals with features that we consider useful, we can hopefully pass these features on to the next generation and eventually the herd. This process takes a long time. Selective breeding relies on the idea that certain characteristics are originally determined by an animal's genetic makeup. Of course, some characteristics can be changed during an animal's lifetime, but these acquired changes can't be passed on to the next generation. Only characteristics with a genetic basis have the potential to be passed on. And this means that an individual inherits a combination of characteristics and genes from its parents. A gene is a section of DNA that acts like a code for a set of instructions. Within the DNA molecule, there are 4 smaller units called bases. And in certain regions of the DNA, the genes, it's the combination and arrangement of these bases that determine which characteristics an animal inherits. Through carefully selecting which animals get to pass their genes on to the next generation, we've been able to create animals with characteristics that suit different environments and different purposes. But we can't always predict exactly which genes an individual will inherit from its parents. There's an element of chance involved. With an individual animal having over 20,000 genes, getting the whole combination of characteristics that we want can be a bit tricky, especially when some characteristics can be influenced by many genes. In recent decades, we've developed tools and processes that allow us to directly change one or a few genes so that we can create plants and animals with the combination of genes that produce particular characteristics. The newest tool in our toolkit is gene editing, or genome editing as it's also known. And the most well-known gene editing tool is CRISPR-Cas9, which is often abbreviated to just CRISPR. Although a recent discovery, this technique uses a process that occurs in nature. CRISPR-Cas9 is part of the defense mechanism in bacteria. When a virus inserts its DNA into a bacterium, CRISPR will cut up the foreign DNA, breaking the code. We can use CRISPR-Cas9's ability to cut the DNA to make targeted changes in a genome. All we need to do is tell CRISPR where to cut by using different guides that correspond to the DNA sequence. Once the DNA is cut, it will naturally be repaired within the cell. When this happens, a different base pair might be inserted into the repair area, changing the sequence of the code. This could lead to a gene being switched off, or could change the function of the gene. These changes look very similar to the spontaneous and natural changes that can happen to living things over generations. We can also provide CRISPR-Cas9 with a template so that when the cell repairs the break, it fills the break with a specific sequence of DNA. This means that the result is more predictable than allowing the DNA to repair randomly. CRISPR can also be used to create a break and insert a different piece of DNA. When gene editing is done on an embryo, the changes will determine how that individual develops. These changes will also be passed on to the offspring of that individual and influence future generations.
Gene editing technology will allow us to make changes in cattle and sheep that we either can't do right now or that would take a very long time. Although the genetic change might be small, gene editing can have big impacts on the health and well-being of the animals, such as creating polled or hornless cattle or making them more resistant to disease. We might also be able to increase the overall productivity of the animals or make meat production more efficient, which is also going to make it more sustainable. Basically, gene editing could boost desirable traits or edit out undesirable ones. So it's going to be important to have a broader conversation about the use of gene editing in meat production.
In Australia, the Office of the Gene Technology Regulator regulates the use of gene editing, including its use in farm animals. However, it's also important that the community has an opportunity to discuss how and under what circumstances this technology should be used. What do you think of the use of gene editing in beef and sheep meat production?